Hello students, in the today's session we'll discuss what is arrhythmias, pathogenesis or mechanisms of arrhythmias and types of arrhythmias. Now first let's talk about arrhythmias. Arrhythmias is defined as abnormality in the normal heart rhythm or the heart rate. Now this abnormality in the rhythm of heart impairs ability of heart to pump blood, and thus it can be a life-threatening emergency. So let's first talk about the conducting system of a heart that regulates the rhythm of heart. And a normal heart produces about 72 to 74 beats per minute. That is, a normal healthy heart shows a heart rate of 72 to 74 beats per minute. Now this is the diagram that shows the conducting system of heart. Now there are 5 elements of conducting system of heart: SA node, that is a sinoatrial node, atrioventricular node, bundle of His. Now this bundle of His divides into 2 branches, right bundle branches and the left bundle branch, and further there are Purkinje fibers. Now SA node is a pacemaker and it possesses the property of automaticity. Cells of SA node generate electrical impulses or action potential without any external stimulus, and about 72 to 74 impulses are produced per minute in a healthy adult. Now impulses from the SA node, they sweep over the wall of both the atria. So these impulses, they sweep over the wall of both the atria and the atrias contract. Now the impulse from the SA node reaches the AV node, and from the AV node the same impulse travels to the bundle of His, from the bundle of His to the 2 bundle branches, that is the right and left bundle branches, and finally the Purkinje fiber. Now this produces contraction of ventricles. So from the conducting system it is very clear that the same impulse, a single impulse that is produced by the SA node, that same impulse is conducted to the entire heart, from the SA node to the AV node, from the AV node to bundle of His, from the bundle of His to the 2 bundle branches, and finally to the Purkinje fiber. And that same impulse is responsible for the contraction of atria as well as the ventricles. That means the conduction follows a proper sequence. And in 1 minute about 72 to 74 impulses are produced. And abnormality in the automaticity, that is the generation of impulses, and conductivity of impulses from the SA node to the Purkinje fibers leads to arrhythmias. Now, in order to understand pathogenesis of arrhythmias, it is important to review action potential in normal contractile cardiac cells and action potential in the nodal or the pacemaker cells. As we all know, nodal cells are the cells which possess the property of automaticity. So the one of the nodal cells is the primary pacemaker, that is the SA node. Now impulses generated by the SA node are responsible for stimulating contractile cardiac cells. That means these contractile cardiac cells, they depolarize only when they are stimulated because of the generation of impulse by the nodal cells. So this is the diagram. It shows the action potential of a contractile cardiac cell. On the x-axis is the time, and on the y-axis is the membrane potential. Membrane potential here is the minus 90, and this membrane potential is increasing as we go upward. Now in the action potential there are 4 phases. This is the phase 4 or the resting phase, which is shown in the black color. This is followed by the phase 0, that is the depolarization phase, which is shown in the red color. Phase 0 is followed by the phase 1, that is shown in the green color. Phase 1 is followed by the plateau phase, that is the phase 2, which is shown in the blue color, and phase 2 is followed by the phase 3, that is the repolarization phase, that is shown in the green color. So let's talk about each phase one by one. So this is the phase 4, this is the resting phase, and the resting membrane potential is minus 90 millivolts. Now phase 4 is followed by the phase 0, that is the depolarization phase, and in this phase there is rapid influx or inward movement of sodium ions through the fast sodium channels. Now because of the movement of these sodium ions inside the cell, there is increase in the membrane potential from negative to positive, and there is rapid upswing of action potential or we can say there is a fast depolarization. Now this rate of depolarization, it determines the conduction velocity. Faster is the rate of depolarization, faster is the conduction velocity. And here one thing important is to note over here is this, that the sodium channel blockers, by virtue of blocking these sodium channels, they can reduce the rate of depolarization, they can reduce the rate of phase 0, and therefore they can reduce the conduction velocity. Now this depolarization phase is followed by the phase 1. During the phase 1, there is efflux or a movement of potassium ions outside the cells. Now there is slight or there is movement of only few ions, few potassium ions outside the cells, and therefore there is slight fall in the membrane potential, and therefore this phase is called as the early repolarization. And early repolarization is followed by the phase 2. Now in the phase 2, this phase is also called as the plateau phase. In this phase there is efflux or outside movement of potassium ions, and there is influx of the calcium ions. So potassium ions are moving outside the cells, whereas the calcium ions are moving inside the cell. However, one thing important to note is this, that efflux of potassium ions is balanced by the influx of calcium ions. Therefore there is not much change in the membrane potential, and this is seen as a plateau phase. And there is contraction of these contractile cardiac cells during the late phase 2. Now phase 2 or the plateau phase is followed by the repolarization phase. Now because of the rapid efflux or movement of the potassium ions outside the cells, there is further fall in the membrane potential, and the membrane potential comes back to minus 90 millivolt, that is the resting membrane potential, and this phase is called as the phase of repolarization. And this is followed by the resting phase, that is the phase 4. Now this period is termed as the effective refractory period. Now during the phase 0, phase 1, phase 2 and phase 3, the cells are refractory. That means during effective refractory period, these contractile cardiac cells, they cannot be stimulated, and they do not produce a new propagated action potential. Now this slide talks about the action potential in the nodal cells, that is the pacemaker cells, that is the SA node and the AV node. Now SA node possesses the property of automaticity, and the SA node depolarizes on its own. And there are 3 phases in the action potential. Phase 0, that is the depolarization phase. Phase 3, that is the repolarization phase, and the phase 4, that is the resting phase. Now this is the diagram. This diagram shows all the 3 phases of action potential. Now this resting phase, that is the phase 4, it is shown in the blue color. Now the resting membrane potential is minus 60 millivolts, but this resting membrane potential is not stable, it is highly unstable, and the resting membrane potential gradually keeps rising because of the influx of funny sodium ions and the influx of calcium ions. So this is not stable at minus 60 millivolts, but it is gradually rising. Now when the membrane potential reaches the threshold membrane potential of minus 40 millivolts, there is opening of calcium channels, there is opening of voltage sensitive L-type calcium channels, and there is influx of calcium ions. Now because of the influx of calcium ions, the membrane potential rises, and this phase, that is the phase 0, is called as the depolarization phase. So this influx of calcium ions in the nodal cells is responsible for the depolarization, and therefore important to note over here is this, that calcium channel blockers, they can reduce the rate of depolarization, and they can reduce the automaticity of SA node, and therefore they reduce the heart rate. Now phase 0 is followed by the phase 3. In the phase 3, we can see that there is efflux of potassium ions, there is movement of potassium ions outside the cells, and because of this movement of potassium ions outside the cells, there is again fall in the membrane potential. So this phase is called as a repolarization phase, and this is a phase 3. Phase 3, at the end of the phase 3, the resting membrane potential again reaches to about minus 60 millivolts, and phase 3 is again followed by the phase 4. Now this slide talks about the pathogenesis or mechanism of arrhythmia. Now arrhythmia is caused either due to abnormal impulse generation, or it is produced because of the abnormal conduction of impulse. Now first we'll talk about the abnormal impulse generation. Now abnormal impulses are generated either because of the triggered activity or because of the abnormal automaticity. Now abnormal automaticity is seen in the nodal cells, that is abnormal automaticity is seen in the SA node, whereas the triggered activity or the generation of abnormal depolarizations, this triggered activity is seen in the contractile cardiac myocytes. So when we talk about the abnormal automaticity, it is the abnormal automaticity of the pacemaker cells or the SA node. So SA node or the pacemaker of heart, as we all know, generates impulses at a rate of 72 to 74 beats per minute, and that is why the heart rate, normal heart rate, is 72 to 74 beats per minute. But if the automaticity of SA node decreases and it produces less than 60 impulses per minute, that is heart rate is less than 60 beats per minute, then in that case arrhythmia is termed as sinus bradycardia. But on the other hand, if automaticity of SA node increases and it produces more than 100 impulses per minute, that is heart rate is more than 100 beats per minute, then the arrhythmia is termed as the sinus tachycardia. Now when we talk about the triggered activity refers to the generation of abnormal depolarizations, or we can say abnormal action potentials, or we can say generation of extra action potential or immature action potentials, or ectopic beats. Ectopic beats are the immature beats. So these all are generated, that is abnormal depolarizations are generated during the triggered activity. Now because of the generation of these abnormal or immature beats, there is increase in the heart rate, and because of the increase in the heart rate, it results in the tachycardia. Now this triggered activity produces two types of depolarization. One is the early after depolarization and the other is the delayed after depolarization. Now first let's talk about the early after depolarization. Now refer to this diagram. This diagram shows action potential in a contractile cardiac myocyte. We have already discussed action potential in a normal working contractile cardiac myocyte. There are 5 phases. If you remember, this is a phase 4, followed by the depolarization, that is a phase 0, partial repolarization, that is a phase 1, followed by the plateau phase, in which there is not much significant change in the membrane potential, and plateau phase is followed by the repolarization. Now in the early after depolarization, abnormal depolarization occur either during the late phase 2, this is a phase 2, they occur either during the late phase 2 or the phase 3. So see this I have shown here in the red color. These are the early after depolarization, these are the immature beats, these are the extrasystole, and these are produced either during the late phase 2 or during the phase 3, that is before the completion of repolarization of contractile myocytes, and therefore they are termed as early, that is before the repolarization. So early after depolarization. Now phase 2 early after depolarizations are caused due to the opening of calcium channels because of the increased influx of calcium ions during the plateau phase, whereas phase 3 early depolarization are caused due to the opening of sodium ions due to the excessive influx of sodium ions. And therefore calcium channel blockers and sodium channel blockers, they can be used in preventing early after depolarization. Now when we talk about abnormal delayed after depolarization, now again this is the action potential figure showing the action potential. Now delayed after depolarization, they occur either during the late phase 3. Now this is the ending of the phase 3, so either they occur during the late phase 3 or they occur during the early this is the phase 4, so they occur either the early phase 4 or they occur during the late phase 3. That means when the action potential of contractile myocyte is nearly or fully repolarized. That means since the repolarization has already occurred, and therefore it is termed as a delayed after depolarization. That means extrasystole is produced after the repolarization. Now these delayed after depolarization, they occur either due to increase in the cytosolic calcium, or they occur because of the excessive stimulation of catecholamines, that is excessive catecholamine stimulations. Now these delayed after depolarization, they lead to rapid tachycardia, and these excessive catecholamine release is inhibited by the beta blockers. So beta blockers, they inhibit delayed after depolarization. Now let's talk about the second mechanism responsible for the pathogenesis of arrhythmia, that is the abnormal conduction of impulse. Now abnormal conduction of impulse can be because of the heart block, or it can be because of a very important phenomenon termed as a re-entry. Now the heart block, that is the blockage of conduction. Now this heart block is normally caused because of the excessive vagal stimulation. Now SA node, as we know, they are the pacemakers. So SA node, it produces the impulses. Now the impulses that are produced by the SA node, they are not normally conducted through the AV node. That is, there is AV block, and because of this there is arrhythmias. So this type of arrhythmia is termed as a heart block. Now another mechanism or another phenomenon responsible for the abnormal conduction of impulse is a re-entry phenomenon. Now re-entry is one of the most important causes of tachycardia. Now these are the three diagrams. Now these are the diagrams of the Purkinje fiber. Now let's first talk about this diagram. This is the diagram of a Purkinje fiber. Now there are two branches of Purkinje fiber. This is the branch 1 and this is the branch 2, and this diagram shows normal conduction of impulse. Now this impulse is coming. This is the Purkinje fiber. This impulse is coming. This impulse, or the depolarization wave, this depolarization wave, it gets propagated in the path 1, that is the branch 1, as well as the branch 2 in a normal way. So this conduction is a normal conduction. Now this is the second diagram. Now this is the Purkinje fiber. It has two branches, branch 1 and branch 2. This depolarization impulse or the depolarization wave is coming. It propagates in the branch 1 normally, but when it tries to propagate in the branch 2, there is a block. This block is because of the ischemia. This is an ischemic zone. Now because of this block which is caused due to the ischemia, this block is a unidirectional block. Now this impulse, it cannot move in the forward direction. So this is a problem with the propagation of impulse. Now further refer to this diagram. This diagram shows the actual mechanism of re-entry. Now again, the Purkinje fiber, the depolarization wave, it is propagating in the forward direction. It propagates normally in the branch 1. When it comes to the branch 2, again, because since it cannot move in the forward direction, there is a there is no more propagation of the impulse, and this impulse or the depolarization wave, it stops here. But when we look at this side, this depolarization wave, it travels or it propagates in the branch 1 normally. Further it takes the path 3, that means it propagates in the path 3. Now what happens is this, that this depolarization wave, it re-enters branch 2. It re-enters the branch 2. And why it re-enters the branch 2? Because this block is the forward block. It is not the backward block. So this impulse or the depolarization wave, it can move in the backward direction. It can move from the path 3 towards the path 2. So this wave or the depolarization waves, it re-enters the branch 2 and it continues propagating. It further propagates in the branch 1. And if it does not stop, it will continue propagating in a circular manner. That means it will keep on re-entering the branch 1 and it will continue propagating in a circular manner, and that will result in the generation of extrasystoles, and that is responsible for the tachycardia. Now very important to note is this, that this action potential or this depolarization wave, it is propagating in this branch 1 only because the cells are excitable in the branch 1. If these cells in the branch 1, they become refractory, then this action potential or this depolarization wave, it will not propagate. So one way to prevent this propagation of abnormal wave or the re-entry mechanism is to increase the effective refractory period of the cells which are found to be present in the path 1. So if we increase the effective refractory period of the cells which are present over here, then this depolarization wave will stop propagating on its own. Another important way by which this re-entry can be prevented is by reducing the conduction velocity of this wave. If we reduce the conduction velocity of this wave, then also we can prevent the re-entry. Now this conduction velocity, it can be reduced, it can be inhibited by the use of potassium channel blockers, and the effective refractory period of the contractile cardiac cells in this branch 1 can be increased by the use of potassium channel blockers. So this is all about the pathogenesis of arrhythmia, the important mechanisms, the most important mechanisms which are responsible for the pathogenesis of arrhythmias. Now this slide talks about types of arrhythmias. There are different types of arrhythmias: ventricular arrhythmias, which originate from ventricle, then atrial arrhythmias, which originate from atria, then sinus node arrhythmias, which originate from the SA node, and atrioventricular block, that is AV block, because of the excessive vagus stimulation, that is the heart block. So ventricular arrhythmia, we'll talk about all of them one by one. First, let's talk about the ventricular arrhythmias. These ventricular arrhythmias originate from the ventricle. These are of different types. First type is the ventricular extrasystoles. The pathophysiology of extrasystoles we have already seen, that is early after depolarization and delayed after depolarization. So there is generation of premature ectopic beats. So generation of premature ectopic beats because of the early after depolarization, because of the delayed after depolarization is responsible for, in ventricles, is responsible for ventricular extrasystoles. Then ventricular tachycardia. There is excessive ventricular rate of contraction. The ventricular rate of contraction is more than 100 times per minute. Then ventricular fibrillation. And now in the ventricular fibrillation, there are uncoordinated ventricular contractions, and the arrhythmia is an emergency because the cardiac output falls to 0. Now when we talk about the atrial arrhythmias, these originate from the atria, and the first is the atrial extrasystoles. Now these extrasystoles, or the premature ectopic beats, these are produced in the atria and they are responsible for atrial extrasystoles. Then paroxysmal supraventricular tachycardia. It is the atrial tachycardia in which the rate of contraction of atria is 150 to 200 times per minute. Then atrial flutter. Atrial flutter, here the rate of contraction of atria is 200 to 350 times per minute. Then atrial fibrillation. Fibrillation, the contractions are highly uncoordinated and the atria contracts at a rate of 350 to 500 times per minute, whereas the ventricles, they contract at a rate of 100 to 160 times per minute. So these contractions are highly uncoordinated. Now when we talk about sinus node arrhythmias, it generates from the SA node. There is abnormality of SA node. Now it is of two types: sinus bradycardia, where the generation of impulses by the SA node is at a rate of less than 60 beats per minute. Less than 60 impulses are generated by the SA node per minute. That results in the sinus bradycardia. And when the automaticity of SA node increases to more than 100 impulses per minute, then in that case the arrhythmia is termed as to be the sinus tachycardia. Now coming to the atrioventricular block or the AV block, this we have already discussed once earlier. There is reduced conduction through the AV node, and this is of three types: first degree block, second degree block, and the third degree block. So this is all about pathogenesis or mechanism of action of arrhythmias and the different types of arrhythmias. Now thanks for watching the video, and I hope you must have liked the session on arrhythmias.